How do B lymphocytes produce immunoglobulins for therapy? Imagine your immune system as a well-trained army, ready to fight off invaders. Among its soldiers are B lymphocytes, or B cells, which are like expert snipers. They produce special proteins called immunoglobulins, or antibodies, that help identify and neutralize threats. But how do these tiny cells make such powerful tools for therapy? Let's find out. B cells start their journey in the bone marrow, where they develop unique receptors called B cell receptors. These receptors are actually membrane-bound immunoglobulins, created through a process called gene rearrangement. This means the B cells shuffle and combine parts of their DNA to produce a vast variety of receptor types, each specific to different antigens. This diversity allows the immune system to recognize many different pathogens. When a B cell encounters its matching antigen in lymph nodes or the spleen, it binds to it using its B cell receptor. This binding sends a signal that activates the B cell. Usually, helper T cells assist in this process, providing the second signal needed for full activation. Once activated, the B cell begins to multiply and differentiate into plasma cells, which are the antibody factories. Some of these plasma cells produce initial antibodies called immunoglobulin M, or IgM, which are the first responders. Others migrate into germinal centers within lymphoid tissues, where they undergo processes called somatic hypermutation and class switching. Somatic hypermutation fine-tunes the antibody's ability to bind tightly to the antigen, while class switching changes the antibody type from IgM to other classes like immunoglobulin G, immunoglobulin A, or immunoglobulin E. These different types serve various roles, such as fighting bacteria or viruses in different parts of the body. The germinal center reaction also creates memory B cells and long-lived plasma cells. Memory B cells stay in the body, ready to respond quickly if the same pathogen appears again. Long-lived plasma cells settle in the bone marrow and continuously secrete high-affinity antibodies, providing ongoing protection. In immunoglobulin therapy, scientists harvest these antibodies from blood donors or produce them in labs. They purify the immunoglobulins and give them to patients to provide immediate immune support. This therapy is especially helpful for people with immune deficiencies or autoimmune diseases, offering passive immunity or helping to regulate immune responses. So, B lymphocytes produce immunoglobulins for therapy by recognizing specific antigens, activating, and transforming into plasma cells that churn out targeted antibodies. These antibodies can then be collected and used to help people fight infections or manage immune disorders. It's a remarkable process that turns the body's natural defenses into powerful medical tools.